Have you ever, tried to find like one specific grain of sand, like on a huge beach,
Oh, yeah.
but you have like this like super powerful vacuum cleaner that can like suck up tons of sand, like thousands of grains at once.
Okay. I like where this is going.
And then like it can flag the ones that look even a little bit interesting.
I'm intrigued.
So that's kind of like a hint at what we're going to be diving into today.
Right.
High throughput screening.
Oh, cool.
It's like a super important part of how we discover new medicines.
All right. I'm excited to dive in.
Welcome to the deep dive, where we try to like sift through all the hard stuff to bring you the knowledge that matters and without leaving you lost at sea.
Very nice.
So yeah, today we're going deep into high throughput screening, or HTS for short.
Right.
And I think you can think about it like as the ultimate speed dating event for drug discovery.
Uh-huh.
You want to find the one molecule that might be like the answer to a disease.
Right.
And HTS lets you introduce your biological target to thousands, even millions of potential partners
Yeah.
in a really short amount of time.
Totally.
So our goal today is to give you a clear picture of what this is all about, like why it's so important, and some of the hurdles involved.
Yeah, some of the interesting nuances and challenges involved.
Absolutely. And we've looked at a lot of material to bring you this today.
We have.
So let's get right into it. Like what, in essence, is high throughput screening?
Well, in the simplest terms possible, high-throughput screening is a highly automated process that allows us to rapidly test a huge number of different chemical compounds. And what we're really talking about is putting these compounds into contact with a specific biological target. So this could be, for example, a protein that's involved in a disease and seeing if any of them have the desired effect. For example, blocking the activity of that protein. So the goal is to find those initial hits, the compounds that show promise.
And when you say huge number, what kind of scale are we talking about here? Like it's not just a few dozen test tubes, right?
No, it's not a few dozen test tubes. Before the advent of this kind of automation screening, even a few hundred compounds would be a massive undertaking. It could take months of painstaking work. HGS has completely revolutionized this. We can now screen thousands, even millions of compounds in a fraction of that time. And this massive scale is truly what makes it such a game changer in the early stages of drug discovery. It allows us to explore a vast landscape of possibilities.
Wow. So it's like going from, examining like a tiny little corner of that beach
Yeah.
to being able to scan the entire coastline in like a matter of hours. I
Exactly.
mean, that really puts the high throughput in perspective.
Yeah, absolutely.
So this is really about finding those very, very first clues,
Yeah,
those initial promising leads when you're searching for a new medicine.
precisely. HGS has become an absolutely fundamental technology platform for this early lead discovery phase. It's where the journey of many potential drugs begins.
Okay, so we've got this rapid, large-scale testing to find initial hits.
Hmm.
Now let's get into the how.
Yeah.
Like, how do scientists actually manage to test so many compounds so quickly? I mean, it sounds like a logistical and a technical marvel.
It really is. And the engine that drives HTS is automation and robotics. These technologies are absolutely essential for handling the sheer volume of samples and the reagents involved in these experiments. Robots can take over many of the manual tasks, like precisely dispensing tiny amounts of liquids into hundreds or even thousands of tiny wells on a plate. They also manage the incubation periods, where the compounds are given time to interact with the target, and then they handle the reading of the results of the assay.
So instead of like, an army of researchers with pipettes,
Yeah.
it's like this super choreographed dance of robotic arms and liquid handlers.
Exactly. Automation takes care of those repetitive, high-precision tasks, freeing up scientists to focus on the more critical aspects, such as designing the experiments, the assays, and then analyzing the mountain of data that these screens generate. And speaking of assays, their design is absolutely crucial for a successful HTS campaign.
Assay design.
Hmm.
So this is like about creating the test that will tell you whether a compound is doing what you hope it will do.
Precisely. A well-designed assay needs to be robust, meaning it gives consistent and reliable results, and it needs to be sensitive enough to accurately detect even subtle interactions between a compound and the target. And a really key point that comes up in our sources is that being able to translate our knowledge of how a disease target works at a molecular level into a reliable, high-throughput assay is a fundamental prerequisite for successful screening. You need to be able to clearly and accurately measure the effect you're looking for on this massive scale.
And these assays are usually pretty targeted.
Right.
They're
Yeah.
not just like general tests,
Right.
but they're really zeroing in on like a specific protein or pathway
Yeah.
that's implicated in a disease.
That's right. HTS often employs targeted assays, focusing on specific pharmacological targets molecules in the body that are believed to play a key role in a disease process. By designing an assay that measures how a compound affects that specific target, we can take a much more directed approach to finding potential therapies.
Now, where do all of these like thousands or millions of compounds come from? Are they just like made on the spot, or are they pulled from like a pre-existing collection?
Yeah, the compounds come from diverse chemical libraries. Think of these libraries as vast catalogs of molecules. Some of these molecules are synthesized in laboratories, specifically designed with certain properties in mind, while others might be derived from natural sources, like plants or microorganisms. Each compound in the library has a unique structural arrangement, and the idea is that by screening a diverse collection, you increase the chances of finding at least one hit that interacts with your target in the desired way.
Okay, so you've got the robots running these really carefully designed targeted assays on this huge library of compounds. So what happens next? How do scientists like sift through all of that data to figure out like what the promising compounds are?
That's where the definition of a hit comes in. A hit is a compound that, in the assay, demonstrates a certain predefined level of activity. And this threshold is set by the researchers before the screening even begins, based on the characteristics of the assay and the specific goals of the project.
So it's not just about if a compound shows like any activity.
Yeah.
It has to meet a certain benchmark.
Exactly. And as some of our sources emphasize, when evaluating a potential hit, researchers don't look solely at how strongly a compound binds to the target or how potent its effect is in the assay. Other factors can also be important at this early stage. What's really intriguing is that sometimes these high-throughput screens can throw up completely unexpected results.
Unexpected results, like what do you mean by that?
Well, HTS can sometimes identify novel and unexpected fragments within the screening hits. These are like small pieces of molecules that show a hint of the desired activity. Researchers can then take these fragments and use them as building blocks, combining them with other known molecular structures to explore and expand what we call the structure-activity relationship, how the structure of a molecule relates to its activity. It can open up entirely new and unforeseen avenues for designing even better drug candidates. So the initial hit isn't always a perfect, ready-to-go drug. Sometimes it's a valuable clue.
That's really cool. It's like finding these little molecular Lego bricks that you can then like piece together.
Yeah, it's a good analogy.
But I imagine like with so much data generated from these massive screens, there has to be some challenges in sorting through, the real signals from just like random noise.
Right.
So how do you avoid getting like buried under false positives?
That's a really critical point. Distinguishing genuine hits those compounds that truly interact with our target in a meaningful way from false positives is one of the biggest challenges in HTS. False positives are compounds that might appear to show activity in the assay, but that activity is actually due to some non-specific mechanism or even just an artifact of the assay itself.
Assay artifacts. So like, is that something like where a compound is interfering with the way the assay is read, even though it's not having like an actual biological effect?
Exactly. For instance, a compound might be colored and absorb light at the same wavelength that our assay uses to detect a signal, leading to a misleading reading. Another common issue is off-target effects. A compound might show some kind of biological activity in the assay, but it's not by interacting with the specific pharmacological target we're interested in. It could be hitting some other protein or pathway in the cell, leading us down the wrong path.
So you think you found a great compound,
Yeah.
but it's really just like a red herring.
Yeah.
It's either messing with the test itself or it's doing something totally unrelated.
Yeah.
So how do you weed these out? How do you get rid of those imposters?
Well, researchers employ several key strategies to deal with this. One important approach involves billing and using large databases of reference data. These databases contain information about compounds that are known to be problematic, perhaps they're known to be nonspecific, or to frequently cause false positive results in certain types of assays. By comparing our newly identified hits to this database, we can flag and filter out likely false positives very early in the process. Another effective technique is to include reference signals directly within the assay. These are control samples that help us to identify compounds that are acting non-specifically.
So it's almost like running tests with known troublemakers
Yeah,
alongside your potential hits.
exactly. And for more complex assays, especially those that involve living cells, we often need further confirmation that a compound's effect is indeed due to interaction with our intended target. This typically involves running additional tests with what we call orthogonal readout.
Orthogonal readouts.
Yeah, so that's like using a completely different method to measure a different aspect of the compound's activity to see if you get consistent results.
Okay, so you run your initial screens, you apply all your filters, you've done your orthogonal testing,
Right.
and hopefully you've got this much smaller set of candidates.
Yeah, a much smaller but much more promising set of potential drug candidates.
So what happens to these next?
Ideally, as our sources point out, a good HTS campaign should deliver more than just one lead compound. Having multiple structurally distinct hits gives us more options moving forward. The next crucial step is prioritization. This is where researchers begin to take a much deeper dive into the remaining compounds.
And when they're prioritizing these, what factors do they consider? Is it just about which one was like the most potent initially?
Not solely. At this stage, early testing of what we call ADMETox properties becomes absolutely critical. ADME stands for absorption, distribution, metabolism, and excretion, essentially, how the drug behaves once it's inside the body. Tox, of course, refers to toxicity, any potential harmful effects the compound might have. These early ADMETox studies help us to identify compounds that are not only active against our target, but also have a reasonable chance of being developed into a safe and effective medicine. For example, we might find a compound that's incredibly potent in our initial assay, but if it's rapidly broken down by the liver and never reaches its target in the body, or if it shows significant toxicity in early tests, it would be a much less desirable lead to pursue.
So even at this super early stage, you're thinking about like whether these molecules could be turned into a real medicine for patients.
Absolutely. It's about sifting through the initial promising molecules to find the ones that not only work in a test tube, but also have the right characteristics to be developed into a viable medicine.
Okay, so to recap for our listeners today, high throughput screening is this really incredible tool that uses automation and these really well-designed assays to rapidly test, you know, tons of chemical compounds against specific targets.
Right.
And the goal is to identify those hits, the compounds that show the desired activity. But this is a complex process with a lot of hurdles. Right?
Yeah, it is.
Especially, distinguishing true hits from false positives. And that requires like sophisticated filtering strategies and orthogonal testing.
Yeah.
And then the most promising ones go through even further testing,
Yeah.
including, assessments of their ADMETox properties.
Right.
Would you say that's a pretty good summary?
That's a fantastic summary. HTS is truly a cornerstone of modern drug discovery, enabling us to efficiently explore the immense possibilities within chemical space.
And these hits, as exciting as they are really just the very, very beginning of what can be a really, really long journey to actually develop a new medicine right.
Exactly. The compounds identified through HTS are just the starting point. There's still a tremendous amount of work to be done in medicinal chemistry to optimize these leads, in pharmacology to understand how they work in more detail, and in toxicology to ensure they're safe before they can even be considered for testing in clinical trials in humans and hopefully eventually become approved medicines.
I mean, knowing the sheer scale and complexity of this whole screening process, what does that tell us about the journey that a potential medicine must go through before it ever reaches a patient?
It certainly does. It highlights the dedication and perseverance required in the quest to develop new therapies.
It really underscores just how many steps and potential pitfalls lie ahead.
For sure.
Well, thank you for joining us today on this deep dive into this super fascinating world of high throughput screening. It's truly amazing to see, the level of automation and, all the science that goes into these really early stages of drug discovery.
My pleasure. It's a process that underpins so much of modern medical progress.
So for our listeners out there, we encourage you to think about this approach,
Yeah.
this highly automated approach, for the future of medicine. And maybe even explore some related topics,
Yeah.
like the intricacies of assay development or the field of medicinal chemistry, or what actually happens to these initial hits as they navigate, the long and challenging drug development pipeline.
Yeah, there's so much more to learn.
Absolutely. So join us next time on The Deep Dive as we, you'll just have to tune in to find out what we'll be unpacking next.
That's